Hello, this is Dr. Hanaa Assil, and this is about human alimentary canal, which is the digestive system. And this is the syllabus for the IGCSE combined science. So, you know that the alimentary canal or the digestive system is made up of these parts, and the first thing that you need to know is the names of the parts. So, let's take a look at the names. We put in food into the mouth. In your mouth, we have salivary glands, and then the food goes down that long tube called esophagus into the stomach, and then it comes out of the stomach into the first part of the small intestine, which is called the duodenum. Now, the food stands in the duodenum. While it is there, the pancreas will secrete juices, and the liver will also secrete juices while the food is standing in the duodenum. After that, digestion is completed. The food goes into the ileum, which is the second part of the small intestine, and the digested food is absorbed into the blood in the ileum. Anything that is not absorbed goes into the large intestine or the colon, and then it accumulates in the rectum until it is sent out of the body through the anus. Did we pay attention to the names? Let's try again. Now, what was A? Remember, A is something in the mouth that gives out saliva into the mouth, so these are the salivary glands. What was B? B is the tube that connects between the mouth and the stomach, that is called the esophagus. Then we go into C, which is the stomach. What is D? That looks like a leaf like that. That is the pancreas. Then we said the food will go, we have after D, we have E. What is E? E is the large intestines. What is F? F is the rectum. This is where the undigested food remains until it goes out through the anus. What is H? Remember that we said I and H are both the small intestine. H is the second part of the small intestine, so that is called the ileum. What is I? I is the duodenum. This is where all digestion is completed. Then we have J. J is the liver. Now, we're going to say that the liver secretes bile, and the bile is stored in the gallbladder. Are we now familiar with the names? Okay. Let's go into what we mean by digestion. You should know that digestion is breakdown of large food molecules, which are usually insoluble. We're going to break them up into small soluble molecules, and this will use two kinds of processes that we're going to talk about, mechanical and chemical processes, in order to be absorbed into the blood. For example, the food we eat is usually large chain of molecules. So it could be, if it's proteins, it is chains of amino acids. If it is starch or carbohydrates, it's a chain of glucose units. If it is fats, it's a chain of fatty acids and glycerol. And the point of digestion is these large food molecules, carbohydrates, proteins, or fats, cannot be absorbed into the blood. So we have to break it up into smaller molecules so that it's absorbed into the blood. So digestion is the breakdown of these large insoluble food molecules into smaller water-soluble molecules that can be absorbed into the blood. Now, how do we do digestion? We do digestion by two methods. We do it by mechanical digestion and chemical digestion. The first thing you do to your sandwich is that you bite into it, you chew it with your teeth. This is called mechanical digestion. Mechanical digestion is the breakdown of the food from large pieces into smaller pieces. So you're not changing the molecules, you're not changing the carbohydrates into something else or the proteins into something else, but you are just cutting your sandwich from large pieces to smaller pieces. This is because when you have larger surface area, then the enzymes that will act on it after that will be able to act easily. Now, how do we do mechanical digestion? We said when you put the food into your mouth, the teeth break up the large piece of sandwich into smaller pieces. And then when the food goes into your stomach, the walls of the stomach are constantly moving. So we say this is the churning movement of stomach walls. So all of this is mechanical digestion. We're just breaking it from large pieces to smaller pieces. We have not changed the molecules themselves. But when we do chemical digestion now is the breakdown of large food molecules into smaller molecules. So I am changing the carbohydrates into smaller molecules. I'm changing the fats into smaller molecules. This uses enzymes. So when the enzyme acts on the food, it breaks it up from large molecules into smaller molecules which are more soluble, which can be more easily absorbed into the blood. Do we understand the meaning of these two? Okay. Once you put your food into your mouth, that is called ingestion. So ingestion is taking in food substances through your mouth, putting the sandwich into your mouth. That is ingestion. Now, what happens in your mouth? In your mouth, you chew the food, and it is better to chew the food. Now, why is this good? When you chew the food, the teeth do what we call the mechanical digestion. The teeth breaks down, crushes the food into smaller pieces, so this provides larger surface area for easier enzyme activity. At the same time, in your mouth, you have saliva. The liquid in your mouth is saliva. Now, saliva has two things. It has a liquid, which we call mucus, and when the food is mixed with the mucus, this makes the food softer and easier to swallow. We also have an enzyme. Now you have to know which enzyme do we have in the mouth. We have amylase enzyme. Now what does the amylase enzyme do? It breaks up starch into maltose. Starch is a long unit of glucose. Many glucose units joined together. The amylase will break it up into twos. So maltose is made up of two glucose units. So we're breaking it up from the long chain of glucose, which we call starch, into shorter chains, which we call maltose. So remember that the only enzyme that we have in our mouth is amylase. So the only substance that is digested chemically in your mouth is starch. If your sandwich has proteins or fats, there is no enzyme in the mouth to break down fats or proteins. The only thing that's broken down in your mouth is the starch using amylase enzyme. Now, the teeth, you probably studied this before. These are the names of the teeth. So, at the front of your mouth, you have four sharp teeth, which we call incisors, and these are used to cut the food. So when you cut your sandwich, you cut it with the incisors in front of your mouth. Then there is one on either side of the incisors that is slightly pointed that we call canine. The canine is for tearing the food. So in carnivores, that tooth on both sides, which we call canine, is actually very sharp and very long for lions or tigers because they use it to tear the food. For humans, it's not that pointed, but it is slightly different. Now, in the back of your mouth, you have two premolars and you have three molars on both sides of your mouth. These are for crushing and grinding your food. So all of this will help to break your food from large pieces into smaller pieces that will increase the surface area for easier enzyme activity. So all of this is mechanical digestion. Now your tooth has a strong outer layer which we call the enamel. The enamel is very strong. It can be dissolved only if you have a lot of acid in your mouth. So that is why we try to brush our teeth so that we don't have acid in the mouth, so that the enamel is not dissolved. Otherwise, the enamel is a very strong part of your body. Then the inside parts are dentine and pulp cavity, and in the pulp cavity we have nerves. So if your enamel were to get a hole in it, if the hole goes in through the dentine and to the pulp cavity, then you'll start to feel pain or toothache once the hole reaches the nerves. Otherwise, for a healthy teeth, this is the part of your tooth. Okay, so we took in food and we said that is called ingestion. Now the food has to go down that long tube called esophagus. You should know that when the food goes down your esophagus, it goes through by a process called peristalsis. So peristalsis is the movement of the food down the esophagus or down the alimentary canal in general. Now, what is peristalsis? You should know that in the walls of the esophagus, you have muscles. You have circular muscles and you have muscles in the walls of the esophagus that are longitudinal. These muscles, when the one above the food contracts, the one below the food relaxes, so this helps to push the food down. So actually peristalsis is successive contraction and relaxation of muscles, as if someone is just pressing on your food to push it down. The muscles in the walls of the esophagus contract to push the food down. This is called peristalsis. Then we go to the stomach. So we went down the esophagus, the food went down the esophagus and reached the stomach. Now what happens in the stomach? The juice inside the stomach is usually referred to as gastric juice, and the gastric juice contains two main things. It contains hydrochloric acid, and the acid in your stomach is very important because it will kill any bacteria that may come in with your food. Remember that however clean your food may be, it will still have some bacteria in it. This will be killed by the HCl in your stomach. It also provides up acidic pH for the action of pepsin enzyme. The pepsin enzyme in your stomach requires acidic pH, and this enzyme is an enzyme that acts on proteins. So the enzyme in the stomach is pepsin, or in general, we call them protease. Any enzyme that breaks down proteins is called a protease. The protease that we have in the stomach is pepsin. Pepsin breaks down proteins in the presence of HCl. It needs acidic pH in order to work. It breaks down the proteins into polypeptides. You should know that proteins are long chains of amino acids. When the pepsin breaks them up, it breaks it up into shorter chains of amino acids, which we call polypeptides. Then the food goes into the duodenum. Can you see the duodenum? Now, in the duodenum, what happens? The food stands in the duodenum. Remember, we said the duodenum is the first part of the small intestine. And the pancreas will secrete juices and the liver will secrete juices on the food standing in the duodenum. So the liver, you have to know, the liver secretes something called bile juice. So the liquid secreted by the liver is bile juice. And this bile juice is stored in the gallbladder. Can you see the gallbladder? So remember that the source of bile juice is the liver. It is stored in the gallbladder. Now, bile juice contains several things. One of them is bile salts, and the bile salts, when they go into the duodenum, they will break down the large pieces of fat, large globules of fat, into smaller globules. This is called fat emulsion. So it forms fat droplets or fat emulsion, This is because now we are increasing the surface area, so when the enzyme comes to act on it, it will be able to break it up. Now, which enzyme breaks it up? It's lipase enzyme, and the lipase enzyme comes from the pancreas. You have to remember, lipase is the enzyme that breaks down fat. It comes from the pancreas, and it breaks down the fat into fatty acids and glycerol. Again, we said the pancreas will secrete a juice into the duodenum. Now, actually, the juice from the pancreas has three enzymes that we're going to talk about. It has an enzyme called trypsin, which is very similar to the pepsin in the stomach. It breaks the proteins, long chains of amino acids, into shorter chains, which we call polypeptides. The pancreatic juice also has an enzyme similar to that in the mouth. We said in the mouth you have amylase that breaks down starch to maltose. What if you swallow your food without chewing? The starch still needs to be broken down. So in this case, when it goes to the duodenum, the pancreatic juice, or the juice coming from the pancreas, contains amylase that breaks down starch to maltose. And then we said the pancreas also secretes lipase. So we said the bile salts in the bile juice that come from the liver break up the fats into fat emulsion, or tiny pieces of fat, but it's still fat. Now, the enzyme from the pancreas, which is lipase, this is what breaks down the fat into fatty acids and glycerol. Then the walls of the intestines also secrete, we're still standing in the duodenum, the walls of the intestines secrete enzymes. It secretes maltase that will break up the maltose to glucose. Remember that we had amylase that breaks down starch to maltose. We now have maltase that breaks down the maltose into glucose. So this is the final end product of digestion of starch. Now, we also have some peptidases coming out from the wall of the duodenum. This breaks up the shorter chains of polypeptides into units of amino acids. So that is the end product of digestion of protein. So now all starch is glucose, all proteins are amino acids, and previously we had already changed fats into fatty acids and glycerol. So in the duodenum, all digestion is completed. Then the food goes into the next part of the small intestine, which is the ileum. So in the ileum, what happens? The digested food, which is now glucose, amino acids, fatty acids, glycerol, all of this needs to go into the blood. This process is called absorption. So this happens in the ileum, or in the second part of the small intestine. We have absorption of the digested food into the blood so that it can be sent to all parts of the body. Okay, whatever was not absorbed in the small intestine will go into the large intestine. Now, what happens in the large intestine? Only water and salts are absorbed in the large intestine. The rest of the food, the rest of the undigested food, whatever was not absorbed, is sent out through the rectum and through the anus. This is called egestion. So egestion is passing out of the remains of undigested food from the anus in the form of feces. Remember, this is not excretion, it is egestion. Okay, let's take a look at some questions on the alimentary canal. The table shows the regions of the alimentary canal where ingestion and egestion may take place. Which row is correct? We said, what is ingestion? Remember, we said that ingestion is when we put the food into our mouth. So ingestion happens in the mouth. What about egestion? We said egestion is removal of the undigested food from the body through the anus. So A is our correct answer. Which diagram represents digestion of food molecules in the alimentary canal? Which of these represents digestion? Digestion means I'm starting with a long chain of molecules joined together, and basically I am cutting it up into small molecules. So of course that would be choice A. Can you see that? Which row correctly matches the enzyme to the product? Okay, so he has lipase. Remember, lipase is an enzyme that acts on what? You should remember lipase is an enzyme that acts on fats. It breaks down fats into what? Fatty acids and glycerol. Lipase breaks up fats into fatty acids and glycerol. It doesn't produce amino acids because amino acids come from proteins. Lipase do not break down proteins. The enzymes that break down proteins are called protease, and the end product of protease breaking down proteins is only amino acids. So remember that when a protease acts on proteins, it gives in the end amino acids, not glycerol. Lipase acts on fats and gives glycerol and fatty acids. Animals break down large insoluble molecules into small soluble molecules in the alimentary canal. What is this process? Large molecules into smaller molecules. Is that chemical or mechanical? That is chemical. Chemical what? Chemical digestion, not chemical ingestion. Okay? Which breakdown processes occur inside the cells and which occur outside the cells? Well, large molecules to small molecules for absorption, where does that happen? This happens in the digestive system or in the alimentary canal. That is outside the cells. It's not inside cells. But breakdown of glucose to release energy, remember that this is respiration, and this happens inside the cell. So our answer actually is C. The diagram shows a section through a human tooth. Which part is made of the hardest material? We said which of these is hardest? It is the outer part, which is A, which we called enamel. Okay? In an experiment, an enzyme from the human alimentary canal is found to work slowly at 20 degrees Celsius. What is the optimum temperature for enzymes working in the human alimentary canal? Remember that the optimum temperature for any enzyme, whether it's in the human alimentary canal or anywhere in the cells, is 37 degrees Celsius, which is the body temperature. The diagram shows the arrangement of part of the small intestine and a capillary. What does arrow X represent? What is arrow X? Can you see X? This is small food molecules going into the blood capillaries. Remember we said when the food molecules go from the small intestine into the surrounding blood capillaries, that is called absorption. Which enzymes are secreted from the pancreas? Remember we said the pancreas actually secretes what? It does secrete amylase, and it does secrete lipase, and it also secretes a protease. Remember we said protease is any enzyme that breaks down proteins, and the protease secreted from pancreas is called trypsin. So we have all three of them. So actually our answer is D. What is defined as breakdown of food into smaller pieces without chemically changing the molecule? When we break down food from larger pieces into smaller pieces, that is called mechanical digestion. What is a function of the small intestine? Remember, what does the small intestine do? The small intestine, it doesn't cut food. It provides a large surface area for absorption. We said the small intestine, this is where we have absorption of the food into the surrounding blood capillaries. The diagram shows an experiment at the start and 1 hour later. What we have inside is a starch solution, and it is changing into simple sugars. So if I put starch solution with Q and it changes into simple sugars, what is Q? Q must be the enzyme that breaks down starch. Which of these is the enzyme that breaks down starch? You should know that it is amylase. Remember, we said amylase breaks down starch into a smaller molecule called maltose. The diagram shows the alimentary canal. Which label shows where bile is stored? We said bile is stored where? It is stored in the gallbladder. So which of these is the gallbladder? Remember that B is the gallbladder. A is the liver that makes bile. So we said the liver, which is A, makes bile. But bile is stored where? It is stored in the gallbladder, which is B. The diagram shows part of a human alimentary canal. Where is bile made? We said bile is made in the liver, which is A. Which process is defined as taking substances into the body through the mouth? We said when we put food into our mouth, this is called what? This is called ingestion. Which row shows where digestion occurs? Okay, did we have digestion? Remember digestion, breaking up large molecules into smaller molecules. This occurs where? In the mouth? Yes, we said we have amylase enzyme in the mouth that breaks down starch. In the large intestine, do we have digestion? No. We said the only thing that happens in the large intestine is absorption of water, and then the food is digested out. We don't have any enzymes in the large intestine, so no digestion occurs there. What about in the small intestine? Yes, we said in the duodenum we have enzymes. In the stomach, yes, we have pepsin in the stomach, so our answer is C. What is not absorbed from the alimentary canal into the blood? Which of these is not absorbed? Remember, we can absorb glucose, mineral salts, vitamins. Remember that fiber is something that is not digested and it is not absorbed into the blood. Digestion can be defined as breakdown of what? Where is definition of digestion? Large insoluble molecules. Now, first of all, it has to be large insoluble molecules. Two small soluble molecules. So that is digestion. So that is A. The graph shows the effect of pH on the activity of 4 different enzymes. Which enzyme is most active in the stomach? Well, let's see. The stomach, we said, has HCl. What was the pH in the stomach? HCl has a pH of about 2. So the enzyme that is active in the stomach needs a pH of 2. So that is enzyme A, which we know is pepsin. In which order does food pass through parts of the alimentary canal? Well, you should know the order. So he's saying esophagus to colon to small intestine. That's obviously wrong. Small intestine to esophagus? No. Small intestine to rectum to anus. That's a correct order because we said small intestine to large intestine to rectum to anus. The others are wrong. A healthy person does not eat for several hours, but then has a meal rich in carbohydrates. Which graph shows how the person's blood sugar level changes after the meal? Remember, when you eat something that has carbohydrates, the enzymes break up the carbohydrates to glucose. So the level of glucose in your blood rises. And then if you are a normal person, the body adjusts itself so that the level of glucose returns back to what it was normal later on. So this applies to which graph? Of course, that applies to graph A. Figure 1.1 is a diagram of parts of the alimentary canal and associated organs. Identify the part labeled X. What was X? X is something that hangs over the stomach. Remember, X is the liver. State 2 functions of hydrochloric acid in the stomach. Remember, we said the stomach has hydrochloric acid. What is the function of hydrochloric acid in the stomach? First, it kills bacteria coming in with the food. And then it provides the acidic pH that is needed for the pepsin enzyme to act on the protein. So these are the two functions of hydrochloric acid. Name the two types of digestion that occur. What were the two types of digestion that we talked about? Chemical digestion and mechanical digestion. Fats, carbohydrates, and proteins are made up of large molecules. They have to be broken down by chemical digestion into small molecules. Name the areas of the alimentary canal where we have chemical digestion. Remember, we said chemical digestion is the one that requires enzymes. Where do we have enzymes in the alimentary canal? You have an enzyme in your mouth, and you have an enzyme in the stomach, and you have enzymes acting in the duodenum. But where do we have absorption? We have absorption in the second part of the small intestine, which is the ileum. This figure shows a diagram of the human alimentary canal. The acidity and alkalinity of some of the parts are shown. So he's saying your mouth is about neutral, your stomach is acidic, and your duodenum is alkaline. During digestion, food is broken down by mechanical and chemical. Explain the meaning of the term chemical digestion. Okay, what is meant by chemical digestion? We said it is the breakdown of large insoluble molecules into small soluble molecules. And we use enzymes to do that, and we do that in order to have molecules that are easily absorbed into the blood. The figure shows a graph of how the activity of three different enzymes varies with temperature. Complete the sentence. Curve which one is from the duodenum. Okay, these are curves that show temperature, and we said enzymes will need what kind of temperature? Enzymes need a temperature of about 37. So which of these has optimum temperature about 37? That is curve A. Can you see that curve A has an optimum temperature of about 37. So this is the enzyme that will work anywhere in the body, whether in the duodenum or in anywhere in the body. Figure 1.3 shows a graph of how the activity of three different enzymes varies with pH. Use the information to decide which curve shows the activity of an enzyme in the duodenum. Now we're talking about pH. What is the pH that we need in the duodenum? Remember that we need alkaline or slightly alkaline pH. That means a pH of what? Of 8. So the graph is F because its optimum pH is 8. So that would be the enzyme that works in the... Okay, so I hope you were able to understand this chapter, and thank you for listening.